Thanks for inviting me here to speak, Rook, and thanks to all of you for staying on a Friday afternoon for this talk. I'm going to start in a familiar place, which is the idea of evolution as climbing a mountain, adaptation as a climbing algorithm, but with a different twist that reflects my interest in the role of mutation and mutation biases in evolution. So imagine a climber on a rugged landscape. A human climber is going to scout out a path to the peak, but for an analogy with evolution, we have to prohibit foresight. So let's imagine that there's a blind climber, or the climber is climbing in the dark, or maybe it's a robotic climber, and it's operating just by strictly local rules. And suppose that our robotic climber moves by a two-step procedure of proposal and acceptance. So in the proposal or introduction step, the climber reaches out to grasp a nearby handhold or foothold, and each time this happens, there's a chance that the climber will commit to the second step, commit to that point of leverage and use it to move. All right. So if we impose a bias on this second step so that higher points are more likely to be chosen, this will cause the climber to ascend, to go upward. But we could also impose a lateral bias on the proposal step. Suppose that our climbing robot has longer or more active limbs on the left side than on the right side, and so it tries more points of leverage on the left. The joint probability of proposal and acceptance is going to be greater on the left, so the trajectory of the climber will be biased not just upwards, but also to the left. Now, on a rough landscape, you always get stuck on a local peak. So in this case, the climber will tend to get stuck on a peak that is up and to the left of the starting point. If the landscape is a smooth hemisphere, like what's shown here, then the climber will just spiral up to the left until he reaches the summit, and it won't really matter which direction of spiraling. And you could also imagine something like a continuously changing landscape where adaptation can just keep happening, and in that case, the climber is going to keep proceeding in that favored direction until further change in the favored direction is limited in some way. Now, this metaphor of climbing Mount Probable is just a metaphor or analogy for evolution. It seems to give some insight about what might happen if evolution is this dual process of biases in the introduction of variation and biases in the reproduction of variation. But also the metaphor could be misleading, right? We don't want to really just trust it. The way to find out if it's misleading or not is to make a model in population genetics. And the best place to start with a model, if you're trying to demonstrate some principle, is with the simplest case. In the simplest case, the climber just has two choices, going up and to the left or going up and to the right. And for each one, we can assign a mutation rate and a selection coefficient. Now, if both the mutation rate and the selection coefficient are favoring going to the left, then that's obviously the choice that evolution will favor. The non-trivial case to consider is the one that's shown here, where going to the left, it has a higher mutation rate as shown by the thicker arrow, but going to the right takes you further up in fitness. So what will happen if we simulate evolution according to this kind of model? The question that we want to ask is, how often will the population go to the left versus going to the right, and how will that be influenced by conditions? So results of a model are shown here. The bias in outcomes, going to the left versus going to the right, the mutationally favored outcome versus the more selectively favored outcome, is shown as a function of the mutation bias. And as you can see, all the lines are going up for whether it's low mutation supply or relatively high mutation supply. And whenever these lines go above 1, that means the bias in outcomes is favoring the mutationally favored outcome. Now, in this talk, I'm mostly going to be talking about evidence and about sort of higher level concepts. I'm not going to spend a bunch of time on mathematical theory. I'm just going to present one mathematical result in the next slide, and then provide a slide with some citations to the relevant literature for those who are interested in population genetic theory. So the simple mathematical result is for the limiting case that's shown by this dashed line. As you get toward small un, or as we sometimes say, small mutation supply, you reach this regime called origin fixation dynamics, where the outcome of evolution can be calculated just by multiplying a rate of origin and a probability of fixation. And so if you're considering a ratio of two types of changes, you just take one origin fixation rate, a rate of origin n times mu, times the probability of fixation relative to the other one. And in this case, these n's are going to cancel out, and if we can assume that the probability of fixation pi is proportional to the selection coefficient, as it is for Haldane's 2s, for instance, then the ratio between these two outcomes is just a ratio of mutation rates and a ratio of selection coefficients. That's this dashed line, and even though that seems like a really simplistic mathematical formula, it actually works when you're in the low mutation supply regime. And then as you get into higher mutation supply, things like clonal interference become important. Now, I don't want to focus on theory, but again, just to give some review, this model of one-step adaptation with two choices comes from Yampolsky and Stoltzfus, 2001. This paper and some recent work from Park and Krug address that same model, or a very topologically similar model. There's a very interesting model from Gomez and colleagues on rate of adaptation with two competing traits. I'm going to talk about this work from Kano in a few minutes. And there's several papers that focus on adaptive walks on complex landscapes that are very interesting. So that's a sampling of some of that theoretical literature. For the rest of my talk, what I'm going to do is focus not on the population genetics theory, but on two questions. How does mutation bias influence adaptive steps, the empirical question? And secondly, what are the broader implications for evolutionary theory? And what I'm going to argue is that there is now abundant evidence that biases and mutation influence the steps in adaptation, and this has important implications for a number of issues in evolutionary biology today. So, mutation bias steps in adaptation. Let's start with some evidence from adaptation in the laboratory. So, McLean and colleagues took the bacterium Pseudomonas aeruginosa, and they repeatedly adapted it to the antibiotic rifampicin, again and again and again, separate cultures. In this system, changes tend to appear in the RPO-B gene encoding an RNA polymerase subunit. And McLean and colleagues measured the frequency with which different variants emerged as the result of adaptation. They measured selection coefficients, and for 11 of the variants, they measured mutation rates. And the relationships of those three variables are shown here. This in the middle is the frequency evolved versus the mutation rates. So they measured about a 30-fold range of mutation rates for these single nucleotide mutations in the RPO-B gene. And there's about a similar range of rates of evolution, and there's a good correlation between the two. And this doesn't happen because there's some relationship between mutation rate and fitness. There isn't any. There's also not very much effect of fitness, and this might seem very surprising, but actually the selection coefficients here are so large that the probability of fixation starts to saturate near 1. So there's not a lot of differentiation in the probability of fixation for these beneficial alleles that are so beneficial. Now, if we had more data, we would expect to see a positive relationship here between frequency evolved and the effect on fitness. Okay, so what about another laboratory study that I think is really interesting from Alex Kausa and colleagues from 2015, and what they did was repeatedly evolve. This is again resistance to antibiotics in the laboratory. They repeatedly evolved resistance to cefotaxime using different strains of E. coli, and 2 of them were mutator strains that had a quite different spectrum of mutations than the wild type. So the mut-T mutator favors AT to CG transversions, and the mut-H favors transitions. And when they repeated this experiment of adaptation to cefotaxime with different parents, what they saw was that the progeny of the mut-T parents, so this is a histogram here showing the occurrence of different specific mutants in a particular gene, all right? So this bar goes up to 19. That means that there were 19 different cultures where a mut-T derived strain adapted to cefotaxime by this specific change here. And there were 2 of them from the mut-H parent and 1 of them from the wild type parent. So this box here is all blue because the mut-T derived strains predominantly adapt by AT to CG transversions. And the redness here is all over here is that because the mut-H derived strains tend to adapt by transitions, although they also sometimes have transversions or other mutations. So what we're seeing here is a striking correspondence between the spectrum of mutation specific to particular strains and the spectrum of adaptive changes. And this raises the question of how what is the general effect of the mutation spectrum on the changes implicated in adaptation in some kind of larger types of experiments or in nature? And by the way, when I say mutation spectrum, I just mean a distribution of rates for different types of changes. So it could be for 6 different reversible nucleotide mutation rates, or it could be for some other kinds of changes. So what Kano and colleagues did in a paper that came out last spring was to define a coefficient of mutational influence, beta, with an expected range from 0 to 1. It's just an exponent here on the effect of the mutation rate, on the expected number of changes to a codon that change to a different amino acid. So this is a formula here that's designed to look at missense changes. And from prior work, we kind of expect that beta is going to range from 0 to 1. So when we're in that low mutation supply regime, like in the origin fixation regime, we expect a proportional effect of mutation biases or of the mutation spectrum, and so beta will equal 1. And when there's standing variation at the other extreme of mutation supply, if all the alleles relevant to the outcome of evolution are already present in the starting population, then selection is just going to pick the winner. Mutation bias won't have anything to do with it. And we might see some intermediate regimes then for intermediate conditions. So after setting up this approach, Kano and colleagues then did some simulations to validate it. So we see for simulated results this a coefficient of mutational effect beta as a function of mutation supply. It's very noisy here, so just focus on this line in the center that shows the mean behavior, which is going from a beta equals 1 at low mutation supply and down to a roughly beta equals 0 for a relatively modest or high mutation supply. So these simulations provide some confidence that beta can be inferred from realistic data and that the inferred value is gonna tell us something. And then, sorry for this black blob on my slides here. Then they applied this approach of inferring beta to 3 different data sets. So there's an experimental data set of adaptation of E. coli to temperature stress. There's a combined data for yeast from several different experiments involving adaptation to high salinity, low glucose, and so on. And then there's a large set of changes that have been observed clinically in Mycobacterium tuberculosis that are implicated in resistance to antibiotics. So in each case, they took this list of changes implicated in adaptation and expressed it as a spectrum of frequencies, and then asked how well that corresponded to the spectrum that was predicted from mutation rates. And the result shown in this column is that in all 3 cases, they infer a value of beta that's not significantly different from 1, from proportionality, and it is significantly different from 0. And in fact, in these studies, there's a very close match between the spectrum of individual nucleotide mutations, so these are the individual types of nucleotide mutations that occur in adaptation, and the spectrum of mutation rates that is estimated from completely independent mutation accumulation experiments. Okay, so I've shown you some evidence from experiments with microbes and from this case of clinically relevant adaptation in Mycobacterium tuberculosis. Is there evidence for this kind of effect more broadly from animals and plants and in a broader set of natural situations? To address this question, we can look at some cases of adaptation that have been traced to the molecular level. And over the past 10 or 15 years, there's been some really great, really remarkable examples that have begun to appear in the literature. I wish I could talk about all of these for many, many minutes, because each one of them represents many, many years of work. Like this one right here is showing, this is about cardiac glycoside resistance. On the left here, we have a phylogeny for a bunch of different insects from 3 different orders. These are columns from an alignment of the sodium pump ATP alpha 1, and it shows changes that are phylogenetically associated with 2 traits here. So the species that are shown in yellow, so I'm sorry, I didn't tell the whole story here. So this ATP alpha sodium pump is targeted by some naturally occurring glycoside toxins, including both the cardiac glycosides that are produced by amphibians and the cardinalides that are produced by members of the dogbane family. So probably you've heard of the milkweed case. Milkweed produces this milky sap. It's toxic to most insects, but the monarch butterfly larvae can feed on milkweed, so they're resistant to this toxin, and indeed they sequester it in their tissues so that the adult butterfly uses it as a defense mechanism, making it noxious to predators. And all of the species shown in yellow here actually do that. They sequester the toxin. The gray ones just consume it and don't sequester it. So we have some changes here that are associated with that phenotype, and the ones that are shown here in this table in bold. The ones that are shown in bold here that are associated with the yellow strains have actually been shown by genetic engineering experiments to confer glycoside resistance on the sodium pump at the molecular level. So that's one example of a data set of where phenotypic adaptation has been traced to specific molecular changes. And there's a bunch of other ones shown in this table. In the next slide, I'm going to talk about a meta-analysis of these cases. So this represents spectral tuning of photoreceptors. This is another illustration of cardiac glycoside resistance that involves animals here that involves reptiles mostly, amphibians and reptiles instead of insects. This is representing benzimidazole resistance in worms. Altitude adaptation by hemoglobin changes in birds, tetrodotoxin resistance, and a couple of other cases here. So one kind of bias that we can look for in these cases is transition-transversion bias in nucleotide changes. So most organisms have a several-fold bias of this type. So if we consider the 4 nucleotides here, there's 12 different ways to change them. 8 of them are the transversions shown in red, 4 of them are the transitions shown in blue, and so if they all happened at the same rate, we'd have a 4:8 or a 1:2 ratio of transitions to transversions. But in fact, the ratio is not 1:1, it's often more like 1.5 or 2 or 3 or 4:1 in different organisms. And we can look for this kind of mutation bias in parallel adaptive changes like the ones that were implicated in cardiac glycoside resistance. So what we're doing here is comparing to a null model. The null model says that outcomes are determined by selection only based on fitness effects, regardless of which forms are more or less likely, and the alternative is that the mutational changes that happen more often by transitions because of this mutation bias are going to happen more often in adaptation. And this paper has a full justification for that, using that kind of null model. And by the way, the results I'm about to show you are conditioned on parallelism. So not only are we looking for these changes that have been reported as being associated with adaptation in cases like cardiac glycoside resistance, and have been verified by genetic engineering experiments, they have to have happened at least twice in 2 different species or in 2 different separate populations. So here's a table showing for the cardiac glycoside case and for 9 other cases, the number of paths and events of transitions and transversions. So the way to read this, for instance, starting with the cardiac glycoside case, is that there's 4 different transition changes, missense changes to that ATP alpha-1 sodium pump that can confer resistance and have happened at least twice in different species. So all of these numbers here are at least 2 or more. So these 4 pathways happen 4, 4, 2, and 2 times. There's 4 possible transversion pathways. They happen 7, 2, 2, and 4 times. So here's a change that actually happens 7 times in that phylogeny that I showed you earlier. And if we add up all these numbers, what we see is a 28 to 27 ratio of paths and a 132 to 99 ratio of events. And remember, the null expectation is 1 to 2 if there's no bias here. So here we're seeing a 2-fold excess over the null, and here we're seeing more like a 2.5-fold excess over the null. So now there's more evidence that I could talk about. So there's this really interesting case that goes into more detail on CPG bias in altitude adaptation. Sorry, this citation is not supposed to be in here. This is just supposed to be about stores here. or this specific analysis of transition bias in Mycobacterium tuberculosis data sets, but this will have to be enough for now because time is limited. So I just want you to notice that most of this work is very recent, just in the past 5 years. So what are the broader implications of this influence of mutation bias on the steps that happen in adaptation? That's the topic for the rest of my talk. So I'm going to shift gears away from talking about these details of data sets, away from talking about any specifics of population genetics, to broader implications. And because I don't think of this work on mutation bias adaptation as a narrow topic. I think about it the way that Max Delbrück, the physicist who was one of the founders of molecular biology, thought about bacteriophages. So in physics, the hydrogen atom, which has one proton and one electron, that's the simplest atom. It's the simplest complete system that we can use to probe the fundamental laws of matter. And at the dawn of the molecular biology revolution, Delbrück, the physicist, chose bacteriophage as, he called it the hydrogen atom of biology, or sometimes he said the hydrogen atom of genetics, because it's the simplest complete system that you can use to interrogate the basic laws of genetics or biology. In the same way, the empirical demonstration of mutation-biased adaptation that I just went through using molecular examples might not seem very glamorous, okay? And transition bias is definitely not glamorous. There's nothing fascinating about transition bias, okay? The point is we can use this to show something that hasn't been demonstrated conclusively in 40 years of evo-devo. That's why I've included this quotation here. Cases of adaptation traced to the molecular level are the simplest systems with which to interrogate the influence of biases and variation on the natural process of adaptation, and the results prove that biases in the generation of variation can be effective. They can have a large, direct, and predictable effect on the changes involved in adaptation. Or to put this in a different way, the demonstration of this kind of causation gives us a warrant to draw a green arrow like this connecting cause and effect, connecting tendencies of varigenesis with tendencies of evolution. And by the way, I sometimes use this word varigenesis because mutation is too narrow. It's important to use language that implicates developmental bias as well, as we'll see in a moment. So we can specify this kind of causation using formal models in population genetics, and we have abundant evidence that it has an important influence on the steps that take place in adaptation, not just in the laboratory, but also in nature. And what that means is that this is a new tool that we can have in our toolbox as evolutionary biologists, a theory that we can use to generate hypotheses that relate the details of mutation and development that cause tendencies of varigenesis with patterns in evolution that might be caused by tendencies of evolution. So what do I mean by that? Well, on the left here we have any internal aspects of mutation and development that cause propensities in the generation of variation, given whatever background conditions are operating. And on the right, we have some things to explain that are outcomes of tendencies of evolution. If we see the same things again and again, if there's sort of enhanced parallelism of certain types, that is a thing to explain. And the explanation might involve variation. So to understand the significance of having this kind of theory of biases in the introduction process that makes this link here, it's important to consider what's the alternative, all right? What are the other theories that specify a role for variation in evolution? Theories that we might consider in this place right here. And this isn't a topic we talk about very much in evolutionary biology. We're mostly talking about selection all the time. So the theories that I'm going to mention, I think they should sound familiar to you, but probably you haven't seen them presented in this way before. So if we think about the most important theories, I would say that there are 5 of them. So excluding Lamarckism, just cross Lamarckism off the list. That's a historically important theory, but it's not on my list. So Darwin said that the laws of variation bear no relation to the structures built by selection. In Darwinian thinking, variation just supplies raw materials that selection shapes into adaptation. There's another very old theory, so these are listed in chronological order. Another very old theory is that there are sort of hard limits on selection due to the nature of development that acts in some way prior to selection. A third theory is that recurrent mutation drives alleles to high frequency, or mutation pressure drives alleles to high frequency. That's something that that's a theory that Haldane and Fisher developed more or less as a straw man theory. It's not a very good theory. Then there's the multivariate generalization of quantitative genetics, which specifies a link between tendencies in the generation of variation that are represented in the M matrix and changes in evolution. And then there's finally this theory of biases in the introduction process. Now, some of these are folk theories and some of them are more formalized. So I would say that evolutionary quantitative genetics is the formalization of neo-Darwinism, dealing with quantitative traits that change by infinitesimals, assuming abundant variation. Although the precise implications of this theory for the role of variation are a little bit different from what Darwin and others believed. They believed that if you have infinitesimal variation in everything, then selection can do whatever it wants. That's not actually what the theoretical framework of evolutionary quantitative genetics says, but it's close. And in the history of evolutionary thinking, there are also folk theories like these shown by these green clouds over here that suggest in some way in which mutation or development determines the possible options for evolution, acting in a manner that is logically prior to selection. So Ulberg, one of the founders of evo-devo, one of the early thinkers, said in evolution, selection may decide the winner of a given game, but development non-randomly defines the players. So the theory of biases in the introduction of variation was proposed explicitly as a formalization of this view, okay? That what the theory says is that a process of evolution that depends on events of mutation that introduce new alleles, or events of mutation and altered development that introduce new phenotypes, will be subject to biases in the introduction process by a first-come, first-served dynamic. And what makes this novel and interesting to think about is that this theory didn't exist formally until 2001. It wasn't part of 20th century thinking, it wasn't part of modern synthesis thinking. And even more interestingly, before that time, population geneticists tended to treat the mutation pressure theory from Haldane and Fisher as the formal basis for these sort of internalist or structuralist theories, all right? So it's important to understand history to understand what that theory means. So first of all, it's not a very effective mechanism of evolution because evolution by mutation pressure means transforming a population by many independent events of mutation. So it's illustrated here with this. So the population geneticist John McDonald has an educational website for students, and there's a simulator on there that you can use to simulate evolution by mutation pressure. So that's what's shown over here. And it does it in a very nice way by turning off reproductive variance. So every row here is a lineage, and then there are generations going from left to right. And so it's as if, here's a parent that dies and has one child each generation, and then the next generation has a child and dies, has a child and dies. So, and it stays red. The allele stays red, and then changes to blue here, because there's some rate of mutation to blue. So there's no reproductive variance, so there's no selection or drift. There's only this effective mutation. You don't have to define the theory that way. It's just easier to see if you turn off selection and drift in precisely this way. So over time, the red frequency goes down and the blue frequency goes up. The problem with this theory is that it's not a very effective mechanism of evolution because it requires high mutation rates that are unopposed by selection. But again, remember, this is how population geneticists thought about internalist theories. They thought that they were theories of mutation pressure, and for this reason, they thought that the theories were not very good. In fact, it was widely repeated that those internalist theories were just incompatible with population genetics because they would require such high mutation rates. So for instance, here's a quotation from Gould, who's actually citing Fisher in this passage of his giant book. Since orthogenesis can only operate when mutation pressure becomes high enough to act as an agent of evolutionary change, empirical data on low mutation rates sound the death knell of internalism. Okay, so that's the mutation pressure theory, and let's go back and consider this again in context with these other theories as ways to think about how variation might affect evolution. So the Darwinian theory is not helpful here because the whole point is to say that variation isn't dispositional. Variation is just a source of raw material. It's not a difference maker that can be invoked to explain why one kind of thing happens more often than another. Instead, selection is always the explanation for interesting things in evolution under neo-Darwinism. Now, the mutation pressure theory kind of cuts a hole in this brick wall, but it's not a very large hole because the theory, again, is not a very good theory. It requires high mutation rates unopposed by selection. The folk theory of hard limits says that variation provides a limited set of options from which selection must choose, and that also does not seem like a very useful theory. So, for instance, previously I was talking about transition-transversion bias. Well, there's no constraint against transversions. It's not like transversions can't occur. They just occur at a lower rate. So describing this as a limit on evolution or as a constraint on selection doesn't really make sense. It doesn't actually work that way. I'm not going to say much about evolutionary quantitative genetics, but it does provide, it's a formal theory, it makes specific predictions, it provides an apparatus to connect varigenesis, the origin of new variation, which is represented by the M matrix, with how evolution happens. But it doesn't give us a whole lot to work with in terms of mutation biases in the sense of asymmetric effect on trait values. So if you try to introduce that kind of mutation bias into a quantitative genetics model, there's basically very, very little effect. Now, there are some effects of correlated variation between traits, but it's really a completely different approach that applies to different kinds of traits. And it's not very relevant to what I want to talk about today. I'm not saying that it's not relevant to lots of other things, and there's, of course, great work in this area from people like Thomas Hansen and Günter Wagner and others. Okay, so let's compare. Now, I've just gone briefly through these different theories. Let's just do a little bit of comparison. And again, it's unusual to talk about these theories of variation, and that's really the legacy of neo-Darwinism, of the belief that selection shapes the outcome of evolution and variation merely provides raw materials. We don't usually think about these things. But for someone like me, who's interested in the role of mutagenesis or varigenesis in evolution, these are the major causal theories that I think about. So let's use this kind of framework to compare some of these theories for specific cases. How can we draw on the theories to potentially connect details of mutation and development that cause tendencies of variation with interesting patterns in evolution? So we talked already about patterns of parallelism, where we see an excess of changes that involve transitions or that involve CpG mutations or sometimes GC versus AT mutations. And most of the theories that we listed don't apply here. So neo-Darwinism doesn't apply. There's no hard limits here, so the hard limits theory doesn't apply. Evolutionary quantitative genetics doesn't apply to explaining these patterns of molecular parallelism. We're really down to two theories, the biases in the introduction of variation and mutation pressure. And as I explained, for mutation pressure to be important, you'd have to have high mutation rates unopposed by selection, basically neutral evolution. The weird thing about the molecular evolution literature is that people use this language of mutation pressure, and they tend to assume that effects of mutation require neutrality, even though, again, it's not a very good theory. And it would be better to express these ideas in terms of biases in the introduction process, which is a different way of looking at it. What about the idea of developmental biases in variation? What does that mean? I've hinted already that I think this theory also applies to developmental biases. So let's explore what that might mean. One of the ways that people in evo-devo explain the concept of developmental bias is illustrated on the left here. So the argument that one finds in the literature frequently is that due to the particulars of a developmental system, or due to the structure of the genotype-phenotype map, mutation to the starting genotype is more likely to produce some phenotypes than others. So here, mutation to the starting phenotype P0, or the genotype encoding this starting phenotype, is more likely to produce phenotype P2 than it is to P1. So there's an asymmetry in the mutationally accessible, in the phenotypes of the mutationally accessible genotypes here. Now, this is not a mutation bias. All the mutation rates are the same here, all right? It's a developmental bias that's induced by these asymmetries in the genotype-phenotype map. But if we're thinking about this as theoreticians, we can take these numbers over here and we can just plug them into this model that I showed you earlier. So earlier I showed you this model where we can go to the left or we can go to the right, and there's a mutation rate and there's a selection coefficient. Well, you can just put these mutation rates in here, and there would be a fourfold mutation bias going toward P2 relative to P1, and it would have exactly the same implications in that model. It's just mapping an interpretation onto that model. It doesn't change the structure of the dynamics that it would predict. Because the population genetic theory that I showed you is all about the consequences of a bias. It doesn't say what the source of the bias is. The theory could apply to biases that are mutational in origin or biases that are developmental in origin that are based on a genotype-phenotype map. So one of my favorite genotype-phenotype maps as a molecular evolutionist is the genetic code. So this is the genotype-phenotype map that relates codon genotypes to amino acid phenotypes. And this map induces asymmetries in the accessibility of neighboring phenotypes, like this asymmetry here, going from this GAT aspartate codon. There are two ways to get to glutamate codons and only one way to get to a valine codon. And again, it's not a mutation bias in the strict sense. The mutation rates are all depicted here as being the same. It's a phenotypic bias that's induced by this genotype-phenotype map, the genetic code. But if we want to know what are the implications of this asymmetry, we can just plug it into that same model that I showed you earlier. Now there's a more general implication of this, a kind of long-term implication of this structure of the genetic code. So different amino acids have different numbers of codons. Proline has 4, histidine has 2, tryptophan has 1. And if you think about, in the big picture, think about all of these different relationships between the amino acids. The amino acids with more codons have more mutational paths pointed to them. And so that makes them more findable by an evolutionary process that proceeds by mutation. And one would expect on that basis that they should be more frequent. In fact, this recalls an argument from King and Jukes in their 1969 co-proposal of the neutral theory, where they pointed out this correspondence between the observed frequency of amino acids in proteins and the frequency expected from just generating a random sequence and translating it with the genetic code. And it's mostly an effect. So methionine and tryptophan have 1 codon, serine, leucine, and arginine, which is a bit of an outlier, have 6. These guys here, except for lysine, these guys have 4 here, isoleucine has 3, and these all here down, these ones all down here have 2. So a general feature of genotype-phenotype maps is that there are large differences in the number of genotypes corresponding to different phenotypes, and some genotype-phenotype maps are dominated by particular frequent phenotypes. And more generally, any non-trivial genotype-phenotype map is going to induce asymmetries of 2 kinds that I just described, a local accessibility and a global findability. So the local accessibility is a function of shared contour length. So what I'm showing here is a kind of stylized genotype-phenotype map of how RNA sequences with different folds map into sequence space. So for RNAs, the sequence is like the genotype, and the shape they fold up into is the phenotype. So what this represents is a bunch of different sequences that are just one mutation apart here in this grid that all map to this one fold or phenotype P0. And then there's connected networks here for 2 other folds or phenotypes. So the effect of local accessibility is if we're starting in phenotype P0, a system can evolve from one point to another within this network neutrally. So it could diffuse neutrally within this network, but then occasionally there might be an adaptive shift to one of these other phenotypes, to either P2 or P1. Well, a shift to P2 is intrinsically more likely because it's more mutationally accessible. All right? And that's shown by this shared contour length here between P0 and P2. It's much larger than the shared contour length between P0 and P1. So an RNA with this fold is more likely to evolve to this fold than to this one due to that difference in local accessibility. And then there's also this global effect of findability. So here P2 and P1 have the same number of genotypes, but P0 has twice as many. So in long-term evolution, P0 is more findable for that reason. Larger phenotypic networks have more mutational arrows pointed at them from other regions of sequence space, which makes them more findable. And this argument has been developed very extensively and in a very sophisticated way from Ard Louis' laboratory over the last 10 years. So here they're depicting a model for RNA folds that's kind of set up like the Yampolsky-Stoltzfus model that I showed you earlier, where we have one option going to the left that's not, there's two ways to go up in fitness here, going to the left and going to the right. This one has a higher selection coefficient, but this one has more sequences. There are more sequences that have this fold. So evolving to this one is more likely. And in this 2014 paper, they showed that the RNA sequences the RNA folds that are more numerous in sequence space are more findable, and that this is an effect of mutational accessibility that they call the arrival of the frequent. And then in subsequent work by Kalamudin Dingle, is the first author's name, here, what they did was they generated RNA sequences and folded them, and this the blue dots here show the frequency as a function of rank for the most frequent fold types for sequences of length 100. The dots that are circled in yellow are the ones that are the fold families that are found in nature. So what this shows is that the fold families, there's a striking tendency for the folds found in nature to be the most frequent folds in sequence space. And this has an important implication for structuralist arguments that evolution often, this is an old argument in evolution from structuralists like Darcy Thompson, that evolution somehow relies on intrinsically likely structures. So this is a form of that argument. Folds that are widely distributed in sequence space are in that sense intrinsically likely structures, and they're more findable because of this effect of mutation. It's an argument that has been repeated in the literature of, there's a kind of literature of self-organization, especially referring to RNAs and other molecules, proteins and networks, that's existed in the evolutionary literature for a number of decades, and it was given a big boost by Stuart Kauffman's 1993 book. He brought attention to this type of argument in The Origins of Order. And in the literature, effects that I'm calling findability are typically described in terms of background conditions or in terms of self-organization, rather than being ascribed to a population genetic cause. But this theory of biases in the introduction process provides a formal causal foundation in population genetics for this kind of claim. Again, the theory says that RNA folds that are more widely distributed in sequence space are more findable in evolution because they're more findable by mutation. And empirical results on the right here confirm this prediction. Okay, let's return to this framework one last time before wrapping up. So if we are interested in how the generation of new variants influences the course of evolution, we have some limited options here. What I've argued in the past 20 minutes is that the theory of biases in the introduction process can be applied usefully to a lot of these issues, and in many cases it makes more sense than any of the other theories. Now that doesn't mean it's true. The way to think about this is this is a theory that in a sense has a rigorous basis. Okay, so there's a body of theory that explains how it works theoretically, and there's some experiments that show clearly that this kind of causation happens, and there's some meta-analyses that suggest it's important in nature. So it's in a sense an established principle, and that means we can use it to generate other hypotheses. So we could look at a pattern of parallelism in evolution and consider the possibility that pattern of parallelism is not because selection favors that particular aspect, but it could be some aspect of mutation or development that's influencing tendencies of evolution. So it's a way of generating hypotheses that can then be subject to test. But of course the testing is a lot easier if you have a causal theory. So likewise in the work that I described from Ard Louis' lab, establishing the evolutionary basis of self-organization depended on having this causal theory for the effect of arrival biases. Okay, I'm going to stop here. I want to give a big shout out to my recent collaborators that I love working with. I love meeting with them every week. Brian Gitchlog and Dave McCandlish from Cold Spring Harbor, Alejandro Cano and Josh Payne from the Swiss Federal Institute. So to summarize, mutation biases influence the steps taken in adaptation. And this has sort of immediate importance for contexts where we're looking at short-term adaptation, like the emergence of antibiotic resistance in Mycobacterium tuberculosis, but it also applies to natural cases of adaptation traced to the molecular level, like that case of cardiac glycoside resistance that I explained about, or like spectral tuning. Now, we could look at this as a very narrow molecular topic, and I'm sure some people will, and in that case, maybe it's not very interesting in the sense that transition bias is not very interesting. But what these cases demonstrate is a fundamental principle of evolutionary causation that we can state, like it's stated here. When a process of evolution depends on events of mutation that introduce new alleles, or events of mutation and altered development that introduce new phenotypes, it's subject to biases and introduction by a kind of first come, first served dynamic. And it's this principle of causation that has wide-ranging implications for exploring issues such as the role of development in evolution and the emergence of intrinsically likely structures. All right, thank you for listening. If you have questions, feel free to comment me. Feel free to contact me. And there's a book about this topic that may be useful. Thank you again.